Bismillah Rahman Rahim. My dear cadets of class 9, Assalamu alaikum and a very good morning to all of you. How are you, my dear cadets? I hope you all are fine at your respective homes. I welcome you all to my online class today. This online class will be helpful to you for continuing your lesson because, my dear cadets, you know very well, due to Corona, you cannot attend your regular classes. Now, first of all, let me introduce myself. I am Muhammad Moinuddin Khan, Assistant Professor of Botany, Department of Biology, your biology teacher. At the beginning of today's class, you have to realize what is COVID-19, what is the symptom of COVID-19, and how to protect yourself against COVID-19. Gentlemen, let's see the video about COVID-19.
COVID-19 is an infectious disease caused by a new coronavirus introduced to humans for the first time. It is spread from person to person, mainly through the droplets produced when an infected person speaks, coughs, or sneezes. These droplets can land in the mouths or noses of people who are nearby. These droplets are too heavy to travel far in the air. They only travel approximately 1 meter and quickly settle on surfaces. This is the reason person-to-person spread is happening mainly between close contacts. The exact time that the virus can survive on surfaces is not yet known. So it is wise to clean surfaces regularly, particularly in the vicinity of people infected with COVID-19. Hands touch many surfaces, which can be contaminated with the virus. You should therefore avoid touching your eyes, nose, or mouth, since contaminated hands can transfer the virus from the surface to yourself. When coughing or sneezing, cover your mouth and nose with the bend of your elbow or use a disposable tissue. If a tissue is used, discard it immediately into a closed bin. The most effective way to prevent the spread of the new coronavirus is to clean your hands frequently with an alcohol-based hand rub or soap and water. This will eliminate the virus if it is on your hands. Stay healthy and prevent the spread of COVID-19.
In previous class, we have already discussed about the general structure of cell, different types of cell, different types of organelle of plant cell. Now, my dear cadets, our today's topic is cell division. Chapter 3, subtopics is mitosis cell division. At the beginning of this class, learning outcome. At the end of this class, you should be able to learn, describe the cell cycle, to explain the mitosis cell division, to identify the stage of mitosis and draw the level diagrams of different stages. To explain the importance of mitosis cell division in living organism. Now, my dear cadets, some important keywords: cell, cell division, amitosis cell division, mitosis cell division, cell cycle, karyokinesis, cytokinesis, interphase, prophase, prometaphase, metaphase, anaphase, telophase, centromere, chromatid, chromosome, homologous chromosome, non-homologous chromosome, and so on. Now, my dear cadets, there are two types of organism, unicellular and multicellular, in the world. Multicellular organisms are composed of millions of cells. The life of multicellular organisms starts from one cell zygote. The unicellular organisms like bacteria, yeast, navicula multiply by cell division. So a cell divides to form multicellular living bodies and to multiply unicellular organisms. In 1882, scientist Walter Fleming for the first time observed cell division in the cell of salamander fish. Cell division is essential to all forms of life, and all new cells arise from pre-existing cells. Normal somatic cells contain two sets of chromosomes carrying similar characters. These are called diploid cells. But gametes, it means male and female, contains one set of chromosome. It means half of number of body cell. These are called haploid cell. Now my dear cadets, cell division definition. The process by which one cell divides to form many cells is known as cell division. There are two phases of cell division. First, the nuclear division, which is known as karyokinesis. Karyon means nucleus, kinesis means division, it means karyokinesis. And the second is cytoplasm division, which is known as cytokinesis. Now, types of cell division. Cell division mainly is of three types. Firstly, amitosis, second, mitosis, and thirdly, meiosis cell division. And first, amitosis cell division, which is called direct cell division. In this type of cell division, the primitive nucleus first directly divides into two parts. Later on, a depression is formed at the middle of the cell, and thus the cell divides into two daughter cells. Each daughter cell gradually develops into mother cells. Later on, they may divide in the same manner. This type of cell divisions are found in bacteria, yeast, navicula, etc. So, the process by which a mother cell divides into two daughter cells without any intermediate stage or without any critical stage is called amitosis cell division. My dear cadets, this is the figure of amitosis cell division. This is the mother cell. This mother cell is divided into two daughter cells without any critical process or any intermediate stage, and that's why it is called direct cell division or amitosis cell division. Now, interphase and cell cycle. Interphase. Do you know what is interphase? Yes, my dear. It is a phase or intermediate stage in between the two consecutive cell divisions is known as interphase. In this phase, preparation of the next cell division takes place. DNA replication and synthesis of other materials take place in this period. About 20 minutes to 2 hours time are required to divide a cell normally, but sometimes 24 hours may be taken. Dear cadets, what is cell cycle? Cell division is a continuous process. One mother cell divides into two daughter cells. After interphase again, the next division takes place. The rotation of different phases in this cell division is known as cell cycle. Scientists Howard and Pelk forwarded a cell cycle. According to them, cell cycle divided into two phases. One is cell division and another is interphase. Firstly, cell division. Prophase. Cell division are divided into 5 per stage: prophase, prometaphase, metaphase, anaphase, and telophase. This cell division will take 5 to 10% time only. Now, interphase. Gap 1, synthesize, and gap 2. It will take 90 to 95% time of total cell division. Now, gap 1, subphase. In this phase, enzyme and other materials for DNA are synthesized. Then S, it means synthesis phase. Synthesis of DNA molecule is done. Now, G2, it means gap 2, surface phase. In this phase, nucleus become larger and takes a preparation for next division. Dear cadets, this is the Howard and Pelk's cell cycles diagrammatic figure. Only 5 to 10% time for real cell division, and 90 to 95% time it's only for preparation time. Now, my dear cadets, definition of mitosis cell division. The process of cell division by which one mother cell divides into two daughter cells while the number and characteristics of the chromosomes are same as in mother cell is known as mitosis cell division. On the other hand, the process of cell division by which nucleus and chromosomes of a eukaryotic cell divides only once and the characteristics of daughter cells are same as the mother cell is known as mitosis cell division. In 1879, Schler first observed this type of cell division and named it karyokinesis. Later on, in 1882, scientist Walter Fleming named this cell division as mitosis. My dear guys, where does mitosis take place and why mitosis is it as a equational cell division? Firstly, what does mitosis take place? Mitosis cell division takes place in a all body, it means somatic cell of plants and animals. I repeat, mitosis cell division takes place in all body cells, it means somatic cells of plants and animals. It occurs in all mesoteric cells of plants at the apex of root, stem, and growing leaf. It also takes place for the formation of reproductive organs, as for example, formation of and development of endometrium and gynoecium. Now, mitosis as an equation cell division. In mitosis cell division, each chromosomes equally divides lengthwise along with the centromere into two chromatids, and each part moves toward the nearest pole. As a result, after the division of number of chromosomes of the two new cells remains same as the mother cell. For this reason, mitosis is known as equational cell division. Now, stage of mitosis cell division. There are 5 stages of mitosis cell division. Firstly, prophase, then prometaphase, then metaphase, anaphase, and lastly, telophase. First, prophase. Pro means previous and phase means stage. It means previous stage and it means at the first stage of mitosis cell division. The nucleus becomes larger in this stage. Dehydration starts in the cell, especially in the chromosome. When stained, the chromosomes are visible under the microscope. By the continuous dehydration, chromosomes gradually become shorter and thicker. At the end of this nucleus and the end of this stage, nucleus and nuclear membrane disappear. At this stage, each chromosome split longitudinally, but centromere remains intact. Each part of chromosomes is known as chromatid. Now I recognize prometaphase. Pro means before, meta means middle, phase it means stage. It means before middle stage of mitosis cell division. Prometaphase is the second phase of mitosis, the process that separated the duplicated genetic material carried in the nucleus of a parent cell into two identical daughter cells. During prometaphase, the nuclear envelope breaks down. The breakdown of the nuclear envelope frees the sister chromatids from the nucleus, which is necessary for separating the nuclear material into two cells. Now, some mitotic spindle fiber elongated from the centrosome and attached to kinetochores. Protein bundles at the centromere region on the chromosomes where sister chromatids are joined. Secondly, other spindle fibers elongates, but instead of attaching Two chromosomes overlap each other at the cell center. Prometaphase is followed by the third phase of mitosis known as metaphase. Dear cadets, meta means middle, phase means stage. It means middle stage of mitosis cell division. As first, metaphase begins with the formation of a bipolar spindle body in the region of nucleoplasm. Secondly, in this stage, chromosomes are arranged at the equatorial zone. The centromere of each chromosome remains at the equator and the arms of the chromosomes remain towards the pole. The arrangement of chromosomes at the equator is known as metagenesis. At this stage, chromosomes become shorter and thickest, and finally, the two chromatids become clearly visible and the centromere divide into two so that the two chromatids can be separated. Now, my dear cadets, anaphase. Ana it means ana. It means motion. Phase means stage. It means motion stage of chromatids of mitosis cell division. The centromere of each chromosome divides longitudinally into two. As a result, each chromosome is now completely divided into two identical halves. It means sister chromatid called daughter chromosome. The centromere of each daughter chromosomes remain connected. I repeat, remain connected to the pole on its respective side by a chromosomal fiber. During later phase, the two groups of daughter chromosomes are pulled away from each other and start moving towards the opposite poles. Most probably, this movement is caused by the shortening of the chromosomal fiber. In each group, chromosomes appear V, L, I, or J shape as the centromeres are pulled towards the poles and the chromosomal arms trail behind. Now, my dear cadets, telophase. Telophase, telo means last, phase means stage. It means last stage of mitosis cell division. This is the last phase of mitosis. The two sets of daughter chromosomes reach the opposite pole. Hydration takes place in the chromosomes. As a result, chromosome starts expanding and gradually become thin and long. A new nucleus is organized. My dear cadets, a daughter nucleus is organized at each pole in the parent cell. Each daughter's nucleus has the same number of chromosomes as that of the mother cell. The spindle fibers are also dissolved and disappeared gradually. The two daughter nuclei are identical in structure and characters. They are also exact copies of the original parent's nucleus. At the equatorial zone, gradually a cell plate develops and cytogenesis. It's time for cytogenesis. What is cytogenesis? Cytogenesis is the division of cell cytoplasm into two separate cells. It usually occurs in telophase along with the formation of daughter nuclei after the nuclear division. The process of cytogenesis differs in plant and animal cells. In animal cells, cytogenesis starts by the appearance of a shallow groove or furrow in the cytoplasm at the equator of the spindle. Slowly, it deepens and constricts the cytoplasm and the cell divided into two parts in plant cell. Cytogenesis is accomplished by the formation of fibroblast, and cell plate at the equator of the dividing cell. Now, my dear cadets, importance or significance of mitosis cell division. Firstly, physical growth. The number of cells increased by the process mitosis is multicellular organisms. As a result, growth and development take place in living organisms. Reproduction. Unicellular eukaryotic organisms reproduce by this process, like Navicular, Chlamydomonas, etc. LG, maintains the equality of chromosome. By this type of cell division, all the body cell bear the same number of chromosomes. Now, vegetative reproduction. This cell division helps to reproduce vegetative of an organism. Healing of injury. This process of this cell division helps to heal up the injury, maintaining equality. Maintaining equalitative stability. This process of cell division maintains equalitative stability in the living organism. Now, the silent features of mitosis cell division. Mitosis occur in the body cell. One mother cell divides into two daughter cells. The number and the characteristics of chromosomes of daughter cells are same as the mother cell. The growth and development of living organism due to mitosis. Unicellular organisms increase their number by mitosis. In vegetative reproduction of plants and animals, the number of cells increases by mitosis cell division. My dear cadets, let's enjoy a video about mitosis cell division.
Cell division is required for an organism to grow, mature, and maintain tissues. During the mitotic phase, the cell will undergo mitosis to form two new nuclei, and then divide to form two new individual cells during cytokinesis. Mitosis is the process of dividing the duplicated DNA of a cell into two new nuclei. Mitosis is split into distinct stages. The first stage is prophase. The DNA condenses, organizes, and the classic chromosome structure appears. Next comes prometaphase, where microtubules attach to the chromosomes. This step is followed by metaphase, where the chromosomes align. Metaphase is followed by anaphase, where the chromosomes separate. Finally, during telophase, nuclear membranes reappear around the two sets of chromosomes. Mitosis is now complete. After mitosis, two new cells are formed by a process called cytokinesis. Mitosis is only one part of what is called the cell cycle. For many eukaryotic cells, a cell is duplicated every 24 hours. Most of the life of a cell is spent in interphase. Interphase consists of three stages called G1, S, and G2. G1, or gap1, is the first growth stage of interphase. In G1, the cell grows to nearly its full size and performs many of its specific biochemical functions that aid the organism. Next is the S, or synthesis phase. This is an important stage because it is during the S phase that DNA in the nucleus is replicated. The cell next enters another growth stage called G2, or gap2. It is during G2 that the cell finishes growing. Once the cell has duplicated DNA in the nucleus and two centrosomes have appeared in the cytoplasm, mitosis can begin. For a typical eukaryotic cell, this will last about 8 minutes. During the first stage of mitosis, called prophase, we first see the classic chromosome structure. This occurs through a condensation process. At the same time, protein strands called microtubules appear different from the centrosomes in animals. Finally, the structure found within the nucleus, the nucleolus, disappears. Next, prometaphase begins when the nuclear membrane is broken down. At the same time, microtubule strands or spindle fibers are growing from the centrosomes. These strands attach to a protein structure called the kinetochore. One kinetochore is attached to the centromere of each sister chromatid. Next comes metaphase. During this stage, the sister chromatids align along the center of the cell so that both chromatids face toward opposite poles of the cell. Now the sister chromatids are ready to be separated. This occurs during anaphase through a shortening of the microtubules attached to the kinetochores. Additionally, the poles of the cell move farther apart, causing increased separation of sister chromatids. At the end of anaphase, the sister chromatids have moved to the two ends of the cell. Telophase is the final stage of mitosis. It is here the components of the new cells begin to appear. At this point, the spindle fibers are broken up. A new nuclear membrane surrounds the chromosomes at the end of each cell, and the chromosomes uncoil and return to an uncondensed state. Mitosis is now complete. The formation of two cells is all that remains. Following mitosis, the cell undergoes a process called cytokinesis. First, the cell is compressed by a contractile ring that divides the cell in nearly equal halves. By now, the organelles in the cell have been replicated and are now divided between the two halves of the cell. This includes mitochondria, Golgi bodies, and the rough ER. Plant cells also have chloroplasts. Once split, the two new cells are now fully in the G1 stage of interphase and ready again to begin their growth. Let's watch the process one more time. Mitosis begins with prophase. Notice the DNA condensing into chromosomes during this stage. Microtubules appear during prometaphase, and the nuclear membrane breaks down. Metaphase occurs when the chromosomes are aligned at the center of the cell. During anaphase, the chromosomes are moving apart. The telophase stage is marked by the appearance of new nuclear membranes. This is the end of mitosis. Finally, the splitting of the cell occurs during cytokinesis. The two new cells are now ready to grow and perform their specialized functions.
Now, my dear cadets, it's time for homework. It's a CQ, it means creative question. A and B are normal cell division of living beings. Cell division A indicates our body, but B cell division takes part in sexual reproduction. Cell division C is a special type of division for lower organisms. The first question means knowledgeable question. What is mitosis cell division? Second question, it's complete understanding level. What do you mean by reductional division? Applicable question, draw and label diagram any three of first cell division process mentioned in the stem. And finally, higher order question, higher living beings cannot be grown up without the first cell division A. Analyze it. My dear cadets, thank you for attending my online class. If you have any question and query of my class, you can clarify it having an online session with me at the period of your scheduled class. Thank you for your patient hearing and viewing. At last, stay home because we are passing critical and crucial moment Take healthy food, take necessary rest, and take fresh drinking water. Please don't go outside here and there. Take care of your parents. Pray to Almighty Allah 5 times so that we all are overcome this crucial situation. May Allah bless us all. Thank you once again. Your next class is cell division, second part. It means the meiosis cell division. Thank you.